Dear students, welcome to EPG Pathshala. In this module, we are going to learn about the sequential development of neurons from the stem cells. The learning outcomes of this modules are that you will learn about the brain development, the neuronal stem cells, the sequential development of the neurons, the difference between the embryonic and the adult neurogenesis. So, to begin with, let us get used to the basics of the brain. So, what we see that the brain is a complex tissue which is made up of several kinds of cells like neurons and the glial cells. The neurons are the main cells which provide electrical activity, and the glial cells are the supporting cells which provide nutrition to these cells. The brain develops by neurogenesis. The neurogenesis is seen both in the embryonic and the adult stage. The neurogenesis was initially thought to be occurring only in the embryonic stage, and the earlier concepts were that once the embryonic brain development stops, the adult brain stops to grow. But today there are startling discoveries where people have found that the adult brain also undergoes neurogenesis. So, some of these discoveries have come from the audio radiography, in which the tritiated thymidine has been used to find out the neurogenesis. Besides this, certain other tools like electron microscopy, which was discovered in 1900s, as well as certain markers on the cell surface, have also given an evidence that the neurogenesis can continue beyond the embryonic phase. So, to understand the neurogenesis in these two phases, it is first necessary to understand the development of the brain. If we look at the brain development, it is seen that in vertebrates, the brain development is formed from the ectoderm of the embryo, and from the dorsal side of the ectoderm, neural plate arises, which forms the neural tube by neurulation, and this neural tube has epithelial cells which houses the stem cells which give rise to the mature brain. Now, these stem cells are the pluripotent cells which arise from the normal cells by 4 stages. First is that they develop competence, then they become specific, thereafter they are committed, and then it is followed by differentiation. Competence means to develop the potential to form the stem cells. Specification means that they have developed the competence. However, if there are inhibitory substances around, they may not form the neural cells. When they are committed, in spite of the inhibitory signals, they will form the neural cells. and when they are differentiated, they produce all types of proteins which are required for their maturity. Now, all these changes help to form the various types of neural stem cells, the neuroblasts, the glioblasts, etc. So, the major event in the brain development is the neural tube formation. So, what we see that the embryo has basically 3 layers, the ectoderm, mesoderm and the endoderm. The ectoderm forms the neural plate which invaginates to form the neural tube. The neural tube forms the brain anteriorly and the spinal cord posteriorly, and the epithelial cells of this neural tube houses the neural precursor cells or the NPC, and these help to form the neural stem cells. Now, the whole event of neural tube formation takes place in 2 ways, that is primary neurulation and the secondary neurulation. The primary neurulation helps the neural plate to increase the number and form invagination, and this also helps to form 3 sets of cells which are precursors of the brain, the spinal cord cells and the epidermis of the skin. Besides this, it also helps to form neural crests. They also help to form ganglia. Now, ganglias are the clusters of cell bodies whose axons form the nerve. The secondary neurulation helps the 2 folds of the invagination of neural plate to come together to form the neural tube. Now, this helps to form the posterior portion of the neural tube. Now, if we look at the stem cells which are present in the brain development, then we see that initially that the epithelial cells of the neural tube contain the neural epithelial progenitor cells. Now, these give rise to the neural precursor cells. The neural precursor cells give rise to the neural stem cells, and the neural stem cells give rise to the neuroblasts, and the neuroblasts initially form neurons and then they form glioblasts. Now, we again look at how the stem cells help to form the neuroblasts and glioblasts. In that case, so what is seen that in the epithelial cells which they house the neural precursor cells, and these neural precursor cells give rise to neural stem cells. The neural stem cells can either undergo symmetric division or they can undergo asymmetric division. The symmetric division helps to form cells which are similar of their kinds. For example, the neural stem cell pool will be formed by symmetric division. That means that the neural stem cells will form cells which are like them only. Whereas, in the asymmetric division, they form neural stem cells, but besides this they also form neuroblasts. Now, the neuroblasts can give rise to the neurons, but initially it is in the early stages that neuroblasts give rise to the neurons, but thereafter they are differentiated to form glioblasts, and these glioblasts can give rise to the glial cells. The neural epithelial cells contain the neuronal precursor cells or the NPCs. And these give rise to neural stem cells. The neural stem cells can either undergo symmetric division or they can undergo asymmetric division. The symmetric division helps to enlarge the neural stem cell pool because they form cells of their own kinds, whereas asymmetric division helps to add neural stem cells. Besides this, they also give rise to neuroblasts. In the early stages, the neuroblast gives rise to the neurons, but later on these neuroblasts convert into glioblast and these give rise to the glial cells. The neural tube differentiates to form various regions of the central nervous system. The tubes bulge and constrict at 3 different portions of the brain. This leads to the formation of the forebrain, the midbrain and the hindbrain. And the cells in these regions arrange in different layers to provide different functions, and they are full of neurons and the glial cells. Now, when we look at the different layers, we find that the CNS is mainly divided into 3 zones: the ventricular zone, the intermediate zone and the marginal zone. The spinal cord has only 3 layers which are mentioned above, whereas the cortex is divided into following 6 layers: the ventricular zone, the subventricular zone, the intermediate zone, the cortical plate, marginal zone and the molecular layer. The ventricular zone, which is also present in spinal cord, cerebellum and cerebral cortex, it is the sole source of neuron and glial cells in medulla and oblongata and the cerebral cortex. The subventricular zone is present in the cerebral cortex and it helps to form the white matter, as this portion has it is called a white matter because this portion has only the axon part of the neuron. In the intermediate zone, which is present in the cerebral cortex, the cortical plate is part of the new cortex, which is the new area and which is from where the cells will be, the new cells in the adult neurogenesis will show their migration. The brain development occurs by neurogenesis. Neurogenesis means to generate new cells in the nervous system. Now, it is of 2 types: the embryonic neurogenesis and the adult neurogenesis. The embryonic neurogenesis occurs in the embryo and it progresses from the embryo to fetus through the postnatal period. The neurogenesis occurs in the outermost layer of the embryo, which is called the ectoderm. The ectoderm first forms the neural plate, which folds into the neural tube. The outer walls of the neural tube give rise to the central nervous system by 3 processes: the cell proliferation, cell migration and cell differentiation. The cell proliferation leads to multiplication of cells in the epithelial layer, which increases the cell number within the ventricular zone. Cell migration, in this there is migration of neuronal cells from the ventricular zone to the subventricular zone to cortical plate and further. That means, they move towards the new cortex. In cell differentiation, the newborn cell acquires characteristics similar to the mature cells. The neuronal differentiation which initiates dendrite formation, the astrocyte differentiation, the oligodendrocyte differentiation, any of these changes. So, the human brain grows at a very high rate. After birth, this rate continues to go on at a very rapid rate up to 2 years of age. More than 30,000 synapses are formed during the first few years. Now, these changes help to form areas which will give new function. They help to form new memories which are required for thinking and forecasting. They also enable learning with as new connections are made between old and new neurons. Now, until recently, most scientists thought that after the initial growth of neurons during fetal development and early childhood, there is hardly any growth of neurons in the adult brain. But today, the MRI studies have shown that the brain keeps on developing until around puberty, and all areas do not mature simultaneously. Besides this, there are also regressive events which take place along with the neuronal cell formation, like cell death and pruning of the cell connections. There is also a wave of myelination, in which myelin sheath is developed around the cells. Now, this marks the difference between the brain in the early puberty and the adult brain. So, the myelination happens in the later part or during the puberty. In the adult neurogenesis, as obvious that this will happen in the adult brain, and the stem cells which are formed in the embryonic neurogenesis can either divide symmetrically or asymmetrically. The symmetric division produces identical cells, whereas asymmetric division produces daughter cells which are dissimilar. The symmetric division forms the progenitors which maintain the pool of stem cells. The asymmetric division forms the stem cells as well as progenitors which can form neurons, astrocytes or dendrocytes. Besides this, the microenvironment is also very important. It promotes the formation of these cells, and only small fraction of cells survive to form mature cells of the brain, and rest of them die during the maturation process. These cells, after formation, they migrate to the pre-existing cortical cells, that is to the new cortex. And the maturation process of these cells involves turning the genes on and off, developing dendrites and axons. And they also acquire characteristic electrophysiological characters. The adult neurogenesis has been observed in limited areas of the brain. Accordingly, they have said that the brain has two parts, either it is neurogenic or it is non-neurogenic. In the neurogenic areas, they have seen neurogenesis in areas like hippocampus and olfactory lobe, and they have seen that the microenvironment in these areas are also helpful in the adult neurogenesis. To get into the details, in the hippocampus, the subgranular zone of the hippocampus and the anterior part of the subventricular zone of the olfactory lobe are the ones which show neurogenesis. And cells from the olfactory lobes show migrations, and there are experimental evidences show that the neurogenesis in olfactory lobe is more than what is seen in the hippocampus, and the microenvironment in these areas influence the kind of nerve cells formed. Now, neurogenesis is a complex process. Neurogenesis is a complex process where the neurotransmitters play a very important role in the sequential development of neurons in the cortex from the stem cells. The new neurons which are formed are more excitable than the old ones. The experimental evidences in the cultured cells show that certain crucial changes occur within 2 weeks of their formation. Initially, they have high sensitivity to GABA, that is GABA aminobutyric acid, but later on their sensitivity to glutamate neurotransmitter increases. But increasingly GABA is presumed to act excitatory in the early stages and inhibitory or hyperpolarizing in the later stages, which is something which is very strange, because initially what we see in the adult neurons, that GABA is usually very inhibitory, but in this case we see that GABA initially is excitatory but inhibitory in the later stage. In the initial stages, it is the glutamate which provides the inhibition, and this helps to maintain the excitatory inhibitory balance which is required to create various neural pathways. The excitatory action of the GABAergic neurons help to develop proper morphology of neurons in the developing brain. Now, these changes also affect the plasticity of the brain. Plasticity is a phenomena which is seen in the brain, where the brain synapses can be changed to help facilitate the learning and memory process. Now, all these neurons which are formed are also undergoing differentiation and maturation. The differentiation and maturation of the brain encompasses the newly formed cells mature by several changes, and that leads to differentiation and maturation of the neurons. The neurons develop dendrites which help to form new synapses with the other neurons. This helps in learning, reasoning and memory. To form these synapses, the tips of the axons, which are called as the growth cones, they grow in a straight line towards the other neurons and develop new connections. Besides this, the neurons also have to be myelinated by being covered by myelin sheaths, by oligodendrocytes in the CNS and Schwann cells in the PNS. Now, myelination helps in increasing the speed of electrical transmission. Besides this, the neurons are now specialized to secrete specific neurotransmitters like acetylcholine, GABA, etc. Besides this, there are also regressive developments in which there is pruning of the dendrites and the connections. Now, these help to form different pathways which deliver different function. In the regressive development, apoptosis and cell death is also a part, and this occurs in some neurons, and all these processes lead to developing a mature brain. Besides this, the neurons have also to migrate to different areas of the cortex. And interestingly, that is done when they are guided by the radial glial cells, which make the scaffolds on which the cells migrate. The new neurons of the embryo have the potential to move to all layers of the cells of the brain, that is layer 2 to 6 of the brain, whereas new neurons of the adult brain move only to layer 2 from the layer 1, that is the ventricular zone, that is from the ventricular zone to subventricular zone only. So, the neurogenesis has been marked by several markers. Some of the markers which have helped to identify the neurogenesis are the radio labeled thymidine which were used in the early experimentation, but today scientists have used non-radioactive methods to detect neurogenesis. Some of these are proteins which are present in the intermediate filaments like GFAP, that is glial fibrillar acidic protein, or nestin. These are found in the progenitor cells and therefore, they have been very useful in tracking neurogenesis. Besides this, there are other proteins present on the cell surface such as double cotin, polyciliated neural cell adhesion molecule, neuron specific protein, NEU-N proteins, which will help to identify the neurogenesis. So, if we look at the differences between embryonic and adult neurogenesis, then we see that the impact of neurogenesis is seen in mainly in the cortical layer of the developing brain in the embryonic neurogenesis, whereas cells are added to the pre-existing cortical layer of the brain. So, that means that in the embryonic neurogenesis we are in fact laying down new layers, whereas in the adult neurogenesis we are adding cells to the pre-existing layers. If we look at the quantity of neurogenesis, then in the embryonic neurogenesis we are forming lots of cells, that is the number of cells formed is large, whereas in the adult neurogenesis there are very few cells which are formed. If we look at the regions which are participating in neurogenesis, then in the embryonic neurogenesis the whole brain is involved, whereas in the adult neurogenesis we see only two areas which have been shown to show neurogenesis, that is hippocampus and the olfactory lobe. If we look at the migration of new cells, so in the embryonic neurogenesis, new neurons migrate to the outer layers of the brain using radial glial cells. In the adult neurogenesis, the cells migrate to specific positions within the existing layers of the brain. And if we look at the types of the cells produced, then it is seen that it produces all types of neurons, but in the adult neurogenesis it produces very specific types of neurons. To summarize the lesson, we will see that historically we thought that neurogenesis only occurs in the embryo and does not occur in the adult brain. But today we have evidences to show that neurogenesis also occurs in the adult brain. In fact, it gives plasticity to the brain which helps in learning and memory. To facilitate this process, there are several stem cells which have been identified, like the neural progenitor cells, the neural stem cells, the neuroblasts, glioblasts, which have given the property of forming new cells and help in neurogenesis. The embryonic neurogenesis is very rapid, and in fact, it is the one which helps to form the brain and give it the potential to become a mature brain. But nevertheless, the adult brain also has the capacity to form new cells. Of course, it is limited to certain areas of the brain, that is hippocampus and olfactory lobe. The whole process of neurogenesis is quite complex, and it takes a while before a new cell is formed from the stem cells and it forms a mature cell, which involves cell proliferation, migration, differentiation, etc. And the process of neurogenesis has been identified. Earlier, it was identified using radiolabeled thymidine, but today we have user friendly techniques like looking at the intermediate filament proteins like GFAP or nestins, which have helped to identify the progenitor cells, because these intermediate proteins are very prominent in these cells. Besides this, we have other cell surface markers which have helped us to identify the neurogenesis. Thank you for your attention.